The last piece of the puzzle would be to translate the messenger RNA into a protein. So this is what the central dogma theory is. It's how proteins are developed. It's how structures are formed. We take DNA, we transcribe it to messenger RNA, and then we translate it into a protein. There's specific processes that are associated with each of these conversions. So when DNA is converted into messenger RNA, we call this the process of transcription. When messenger RNA is translated into protein, we call that translation, the process of translation. Okay, and then another kind of name for the central dogma theory is the one gene to one protein hypothesis, but you'll rarely hear me refer to it as that. This is primarily known as the central dogma theory, is what I will use to define this process. Okay, so in the process of transcription, we again are converting DNA from its DNA molecular structure, and then we're simply going to manipulate that structure and transcribe it into messenger RNA through the process of transcription. And what makes this possible, again, is a biomolecule in the form of a protein, which is a type of enzyme, it's a type of enzyme which is a type of protein, and that specific protein or enzyme is going to be referred to as RNA polymerase. What RNA polymerase does is it will start to convert the DNA molecule into messenger RNA. Okay, the process of transcription occurs both in eukaryotic cells as well as prokaryotic cells. So bacteria cells are the primary organisms that will be prokaryotic, and eukaryotic cells, remember, they have a membrane-bound nucleus. We'll talk more about the differences in our, I think, chapter 3 when we cover differences between eukaryotes and prokaryotes. But transcription will take place inside the nucleus in a eukaryotic cell. It doesn't take place inside the nucleus of a prokaryotic cell because there is no nucleus. There's a nuclearid region, but there's no nucleus. And so when transcription takes place, again, you have the formation of messenger RNA molecules, and these messenger RNA molecules have to leave the nucleus for the next process, which is the conversion of messenger RNA into protein, to take place. So just know that in eukaryotes, that once transcription happens in the nucleus, the messenger RNA has to leave the nucleus, and then it goes to a structure known as a ribosome, where it will undergo the process of being translated into a protein. So here's another YouTube video that I think will highlight, kind of what happens during the process of transcription. So we're just going to take a look at this. And this is posted on YouTube by Microbiotic. We can ignore that portion right now.
During transcription, the DNA in the gene is used as a template to make a messenger RNA strand with the help of the enzyme RNA polymerase. This process occurs in 3 stages: initiation, elongation, and termination. During initiation, the promoter region of the gene functions as a recognition site for RNA polymerase to bind. This is where the majority of gene expression is controlled, by either permitting or blocking access to this site by the RNA polymerase. Binding causes the DNA double helix to unwind and open. Then during elongation, the RNA polymerase slides along the template DNA strand as ties to the 3' end of the growing RNA molecule. Once the RNA polymerase reaches the terminator portion of the gene, the messenger RNA transcript is complete.
So you can see in that video how the RNA polymerase is the site where you're going to have the laying down of RNA. You're having the laying down of RNA base pairs. Okay, so we have this development taking place here in the RNA polymerase. And then once that happens, you end up generating an RNA, a messenger RNA strand that is going to leave the nucleus and be used in the process of translation. So again, in the process of transcription, you essentially just have DNA being converted into messenger RNA through the use of that RNA polymerase to lay down RNA ribonucleotides, developing one strand of messenger RNA. And that messenger RNA will then leave the nucleus again, and it will make its way to a ribosome inside the cell, and then that will enable the development of, or at least enable the process of translation to take place. Now, so this is what happens in the eukaryotic cell, but in a prokaryotic cell, transcription is going to take place inside the cytoplasm. So transcription doesn't have to, well, we don't have to wait for transcription to happen inside of, we don't have to wait for translation to happen, because translation, which is the process of converting messenger RNA into protein, takes place outside of the nucleus. Transcription in a eukaryotic cell, that takes place inside the nucleus. But in prokaryotic cells, they don't have a nucleus. So transcription is going to take place inside the cytoplasm. And in the cytoplasm of a prokaryotic cell, you basically just have these protein structures that are called ribosomes that are situated all throughout the cytoplasm. And they will link to the messenger RNA molecules as they're being transcribed from the DNA template. And so in a prokaryotic cell, which is different from eukaryotic cell, you can actually have the processes of transcription and translation occurring at the same time, which makes bacterial cells and other prokaryotic cells much more efficient in generating genes and DNA, because they don't have to take that extra step in getting the messenger RNA to pass through a nuclear pore in the nucleus and then make their way to a ribosome. Everything is already done in the cytoplasm, so it omits an extra step. So this process of transcription is very interesting, as essentially the cells are taking DNA and converting it to messenger RNA, so that way messenger RNA can be translated into a protein. Now, viruses undergo this process as well. Viruses, as I've said before, are not living entities, but in terms of viruses wanting genes to be expressed, viruses will package inside of their molecular components an enzyme that's known as reverse transcriptase. And so this enzyme is pretty essential, because what it can do is it can reverse the process of transcription, and that means that it can take messenger RNA and then use messenger RNA as a template to convert it into DNA. And this assists a virus, because a virus is not its own living entity. It's pretty much a parasite that has to rely on another host cell in order to survive. So if it can convert messenger RNA or RNA into DNA, then it can integrate its genetic material inside of a cell or a host cell. And this is made possible by the idea of this reverse transcription that can take place. So here's a pretty good video that's posted by Thermo Fisher Scientific, which is a science research company, defining what reverse transcription is.
The process of making complementary DNA, cDNA, from single-stranded RNA. The experiment requires RNA, a reverse transcriptase enzyme, a primer, and dNTPs.
So don't worry about what all of this stuff means, but we're really just looking how we're going to convert messenger RNA to DNA.
The RT experiment is also called first-strand cDNA synthesis. The steps are as follows. RNA is first incubated with a primer at 70 degrees centigrade to denature RNA's secondary structure.
And all a primer is it's a very small gene fragment that's going to get the coding process started, or the transcription process started.
This is chilled quickly on ice to let the primer anneal to the RNA. Reverse transcriptase, dNTPs and buffer are added to the reaction. RT reaction is extended at 37 degrees centigrade or higher for 1 hour to allow transcription to occur. RT reaction is heated at 70 degrees C to inactivate the enzyme. The end result is cDNA.
Okay, so this really is what we're interested in for the most part. What this guy is talking about is, kind of a kit that can be used to generate clonal DNA inside of a laboratory setting. In nature, when viruses do this, what they do is they will convert this RNA strand into DNA, and they do it by simply adding that reverse transcriptase. The reverse transcriptase will lay down the proper base pairs, and then that we get conversion from RNA into DNA. Now, there's some viruses that employ the use of this enzyme to use reverse transcriptase to convert or integrate themselves into DNA of the host organism, but not all viruses do that. And when they do this, we end up having viral genes being produced. In regards to viral replication is just that viral replication can take place. Essentially, you can have a virus attack a cell and it will send its viral components through the membrane into the cell, and then those viral components will start breaking down organelles and it'll start breaking down the membrane around the cell, and then it will integrate its cell into DNA. Once that happens, it uses the central dogma theory inside that cell to produce its own copies of its genetic material. And when that genetic material is coded or replicated in the nucleus, then it is transcribed into messenger RNA and then translated into protein. So you have the development of viral proteins. And so here's a video posted by Rufus Rajadori on YouTube that's going to kind of demonstrate the replication properties of a retrovirus known as HIV, which uses the reverse transcriptase in order to replicate.
Targeting HIV replication. The replication of HIV-1 is a multi-stage process. Each step is crucial to successful replication and is therefore a potential target of antiretroviral drugs. Step 1 is the infection of a suitable host cell, such as a CD4 positive T lymphocyte.
So a CD4 positive T lymphocyte is just a blood cell, one of your white blood cells that's attacked by the HIV virus. And once you start knocking out your white blood cells or viruses take them out, then you become very much immunocompromised.
Entry of HIV into the cell requires the presence of certain receptors on the cell surface. CD4 receptors and co-receptors, such as CCR5 or CXCR4. These receptors interact with protein complexes, which are embedded in the viral envelope. These complexes are composed of two glycoproteins, an extracellular GP120 and a transmembrane GP41. When HIV approaches a target cell, GP120 binds to the CD4 receptors. This process is termed attachment. It promotes further binding to a co-receptor.
So the first thing that needs to happen whenever you have a virus trying to infect another cell is the point of attachment. The next step is going to be that of penetration, and then
Co-receptor binding results in a conformational change in GP120. This allows GP41 to unfold and insert its hydrophobic terminus into the cell membrane. GP41 then folds back on itself. This draws the virus towards the cell and facilitates the fusion of their membranes. The viral nucleocapsid enters the host cell and breaks open, releasing 2 viral RNA strands and 3 essential replication enzymes.
so this is penetration.
Protease and reverse transcriptase.
So those are the biomolecules we've been talking about, which are in the form of enzymes.
Reverse transcriptase begins the reverse transcription of viral RNA. It has 2 catalytic domains, the ribonuclease H active site and the polymerase active site. Here, single-stranded viral RNA is transcribed into an RNA-DNA double helix. Ribonuclease H breaks down the RNA. then completes the remaining